ICH M13A highlights. The ICH M13A guideline is for bioequivalence for immediate release, solid oral formulations or dosage forms. And in this video, we will quickly go through the highlights of ICH M13A guideline. The IR formulations may be tablet capsules, suspensions, and others which are given by the oral route. These formulations may contain soluble or insoluble type of APIs belonging to BCS classes of 1 to 4. BCS class 1 and 3 are highly soluble and BCS class 2 and 4 are low soluble, and to formulate these formulations there are requirement of special technologies. So this guideline gives you idea about all the requirements and the key points for bioequivalence of IR formulations. Bioequivalence refers to the comparison of the rate and extent of drug absorption between two formulations, mainly the reference formulation and the test formulation. And the purpose of bioequivalence is to ensure that two formulations of the same drug will have similar effects in the body. The pharmacokinetics are studied in the bioequivalence. IR solid dosage forms include tablet, capsules, granules, powders, and oral suspension, and also powders and granules for the oral suspensions. ICH M13A guideline provides a recommendation for conducting bioequivalence studies during both development and the post-approval phases. That means M13A guideline is for the exhibit stage or pivotal stage and also for the SUPAC level changes. Scope of the ICH M13A guideline is applies to orally administered IR formulations, and the purpose of this guideline is to harmonize technical aspect of study design and the data analysis for bioequivalence assessment for IR formulations. Key aspects of this guideline are study design and data analysis, provide scientifically grounded methodologies. BE assessment focus on the bioavailability and pharmacokinetic, that is PK evaluations for immediate release type of formulations. So this guideline ICH M13A will replace the parts of EMA-BE guideline from 25th January of year 2025. That is from 25th of January this upcoming year, the ICH M13A guideline will replace the parts of EMA-BE guideline. So coming to the dosing instructions for BE studies, when only fasting is required, no food effect clinically. So whenever there is no effect of food and it is clinically justified, then only fasting is required. When only fed, testing will be required, or when only fed, bioequivalence study will be required. Positive food effect or reduced variability observed. When fasting or fed will be required. Food for tolerability, not due to pharmacokinetic reasons. When fasting plus fed required. So fasting and fed is required for high risk products. I will go through these high risk products. What are the high risk products as per the ICH M13A guideline? So this guideline is very clear and very informative. All the instructions are given for the state of the BE study, whether we should go for fasting, whether we should go for fed, whether we can go for any, and when fasting plus fed is required. So always consider whenever you are working on to the high risk products, then fasting plus fed condition will be required for bioequivalence demonstration. High risk product for BE studies. What are the high risk products? Characteristics of high risk products are negative food effect, lower bioavailability in fed conditions. Formulation dependent impact influenced by formulation design or components like lipid based formulations and nanotechnology based formulations. Examples are low solubility drugs, complex formulations like solid dispersion, microemulsions, co-processed drug substances. Whenever you go through or whenever you see the high risk product written in this guideline, that means the modification is there for the API. Lipid-based formulations means these are the formulations in which the lipid excipients are utilized to improve the bioavailability, and that's why these are high-risk products. Whenever there is lower bioavailability because of the feed conditions, that time these are the high-risk formulations. Whenever there is formulation dependent impact influenced by formulation design or components like particle size reduction, nanotechnology, that time there is a high risk for the bioavailability. Examples will be like low solubility drugs in which solubility enhancement technologies are used, techniques are used to make the formulations, and that's why the formulations become complex. Solid dispersion made by hot melt extrusion, solid dispersion made by spray drying, by amorphous solid dispersion technologies, microemulsions, co-processed drug substances. Whenever there are these technologies in the product, then these are the high risk formulations. Now we will see what is standardization for fasting and fed studies. Fasting study is 8-hour fasting before dosing. Water free access will be there except 1 hour before and 1 hour after dosing, and volume of water will be 150 to 250 ml. Fed study requirements: pre-dose meal 30 minutes before dosing, and meal types will be high fat and low fat. High fat meal means 50% of the total calories are coming from the fat. Low fat means approx 500 kilocalories. So this is regarding the standardization and fasting and fed studies. Standardization for high risk products. Fasting plus fed studies required for high risk products and high fat, high calorie meal containing or giving the 900 or 1000 kilocalorie will be the high fat, high calorie meal. Meal composition will be 500 to 600 kilocalorie from fat, 150 kilocalorie from protein, and 250 kilocalorie from carbohydrates. Adjustment for patient tolerability. For high risk products where tolerability is a concern, that time adjustments are given. Implementation timeline for ICH M13A. European Commission 25th January 2025. FDA 31st October 2024. Health Canada implementation in process. PMDA Japan not yet implemented. Swiss Medic implemented on 23rd July 2024. So this is the implementation timeline, and you will see the upcoming product specific guidances for BE studies will be revised. You can also see current actions and updates by the US FDA. FDA has revised many of the guidelines, that is product specific guidances PSG for IR products. Recently I went through the guidances for linagliptin and empagliflozin. Many others are also revised. See, the combination products are also revised based on this ICH M13A guideline. Earlier, for some of the products, the testing for B or B conditions were fasting and fed both, but now only fasting is recommended. So you can go through the product specific guidances and see the revision date for those PSGs, and you will get an idea that after M13A guideline, these product specific guidances are revised for IR formulations. So further updates will be future updates from EMA on how ICH M13 series will replace the existing guidance. The part ICH M13 B and C will also be coming, and that time we will discuss on the same. I have made some videos on ICH M13A guideline. You should go through those videos to have better understanding of this guideline. Thank you for watching the video.